Hi everyone, we're going to continue on with discussing proteins. Specifically, we're going to talk about protein structure. So protein structure is described as four potential levels: primary, secondary, tertiary, and quaternary. And you can see these protein structures on the right-hand side. So the figure at the very top is a linear chain with multiple amino acids bound together through peptide bonds. This is a primary protein structure. This is the most basic protein structure there is. The second picture down below is a secondary protein structure. The secondary protein structure is composed of pleated or pleated sheets and alpha helixes. The tertiary protein structure is the third one down, and here we see a more complex structure with pleated sheets and alpha helixes. And we can see that it's starting to fold a bit. And then the quaternary protein structure is a combination of multiple structures together in a folded pattern. So these are the four main structures of a protein, and they increase in complexity as we go from primary to quaternary. The structure will ultimately determine how the protein interacts with other molecules, specifically substrates and enzymes. The protein structure is also very much linked to the protein function. Now let's begin by discussing the primary structure of proteins. This is the basic structure of a protein, and essentially it's a specific sequence of amino acids in the polypeptide chain. So it's in a linear polypeptide chain with repeated peptide bonds. We can see it in the picture down below on the right. We see a primary protein structure where we have this long linear polypeptide with lots of amino acids strung together. If we were to blow up that amino acid in the bigger picture, we see our carboxylic acid with the amine group, and we see the hydrocarbon as well as the R group. So remember that R group is specific to the amino acid. So the primary structure of a protein is linear, and it's made of repeating amino acids that are linked together by peptide bonds. Now let's discuss how the primary structure is determined. This comes from the DNA sequence, which leads to the RNA sequencing, and then the code for the polypeptide at the ribosome. So let's look at the figure on the left side of the slide. We first of all look at the DNA. And so we see the base pairing of the nucleotides of the DNA, the double helix. We see, for example, that GAC is the code for leucine. And we see another code for an amino acid there, which is TGA. So there's many different codes for many different amino acids. And so this is transcribed into an mRNA sequence for that specific amino acid, which then the mRNA travels to the ribosomes, where it then communicates to the ribosome what protein should be built. And this is where the polypeptide, that linear chain, is developed. And you can see the mRNA, the first sequence, GUG codes for valine, CAU codes for histidine, CUG codes for leucine, and then we have another one as ACU. So this is how that polypeptide chain is developed, which is our primary structure of a protein. It comes from the DNA, which is communicated to the mRNA and then translated into a polypeptide. This becomes the basis for all the other levels of structure that give the protein its chemical and physiological properties. I wanted to put this slide in here to showcase the complexity and really cool features of DNA sequencing. You do not need to memorize this chart. I want you to appreciate it and understand what's going on. So essentially, when we have a DNA sequence, which is translated into an RNA sequence, that RNA sequence then codes for a specific polypeptide chain, which is the primary structure of proteins. So as you can see here, if we have a combination of U and U, so we have UUU and UUC base pairing, this will lead to the creation of the amino acid phenylalanine. If we have UUA and UUG, that's leucine. And so the combinations here are quite, there's a lot of them. And then you can also see the stop messages, which is UAA, UAG, and UGA, and that tells the polypeptide to stop. So that will help in indicating how long that polypeptide chain will be. So it's very cool. This is the DNA to mRNA sequencing that codes for the amino acids we need to create at the ribosome for functionality within the body. Now, I wanted to bring this back to a big picture overview in real life. So this is the Canada Food Guide newly established recommendations as of 2019. And I think it's a great representation, visual representation of what your plate should look like. So half of your plate should be vegetables and fruit. A quarter of it is some sort of protein source, and a quarter of it is a whole grain food. And then make water your drink of choice. So the protein area is really where we're getting our amino acids. So these amino acids, remember, we eat the food, we eat the whole food, we just break it down through the process of digestion and catabolism to our individual amino acids, which go into this idea of an amino acid pool. And then based on the DNA and RNA sequencing, we create our amino acids. Okay, so the genetic code, your specific genetic code requires certain amino acids to build a protein in the body for a specific physiological function. So we eat the protein, that gets broken down, those amino acids go into our amino acid pool. We then get the code from our DNA and RNA to produce which types of amino acids, and we string them together in a polypeptide chain for specific function in the body. If we have an inadequate diet and we do not get the essential amino acids, then our body cannot code and create that polypeptide. And so we'll be in a state of disease or out of homeostasis until we get those essential amino acids to form that polypeptide. Okay, this is important because if we don't have an adequate diet, then you're going to see things go downhill. So you'll see the integrity of your skin, your hair, muscle mass, all the things that neurotransmitters, enzyme activity, all of these are proteins. And so if we're not getting the essential amino acids in our diet, then our body is not getting them to build the amino acids and the polypeptide chains that we need. I will say this multiple times throughout the course, that there are a few things in life that you can control, and one of them is your nutrition. So ensuring that you have an adequate diet composed of all of these things on the plate, here are the visual representation of the plate, is essential. Food pairing is important, not eliminating complete food groups. We need these things in an adequate quantity and quality to ensure that our body is given what it needs, the essential nutrients to repair, build, and maintain itself. Now let's move on to discuss our secondary structure of proteins. These are arrangements of primary structures by hydrogen bonds that form between the backbone atoms of amino acids, not their side chains. So it's the backbone. So remember what the backbone is. It's the hydrocarbons. So what we see happening is hydrogen bonds being formed between amino acids to form these alpha helix or beta pleated sheets with connecting loops. Okay, and we'll talk about this on the next slide. But ultimately, the secondary structure that a chain forms is determined by the primary structure of the chain. So remember, our DNA sequence transcribes into a messenger RNA sequence, which translates into an amino acid sequence, which forms our primary protein structure. And the primary protein structure then dictates what the secondary structure will be. Now let's discuss the alpha helix, which is one of the two secondary structures of a protein. These spontaneously form, and it is a helix that's typically approximately 10 amino acids long. So let's look at the picture of it on the left-hand side. We can see the entire diameter of this is approximately 10 amino acids. We can also see that there are dashed lines which indicate hydrogen bonds, which are formed between the carbon double bond to the oxygen, the carbonyl, and the amine group of the subsequent amino acids. Typically within one turn, there is 3.6 amino acids per turn. And when there is the amino acid proline, this ends the alpha helix structure. And this is because it causes a bend in the structure. So that is usually always at the end. So this alpha helix forms a 3D helical cylinder with the side chains projecting outwards. There's no space in the cylinder and it is extremely strong. So an example of an alpha helix secondary structure is your fingernails and your toenails. These are both in the alpha helix secondary structure of a protein. They're extremely strong, as you can tell by just touching your fingernails or your toenails. Now let's talk about the secondary structure, specifically the beta-pleated or pleiated sheet. Here we have hydrogen bonding, which is between the adjacent chains. We can see it between the amine group and the carbonyl group of the subsequent amino acids on the adjacent chain. We can see that through the lines on the picture on the left that are highlighted in yellow. So several chains can be involved. And what we find is that these pleiated sheets are 3D structures that are formed, and they look like crinkles, like corrugated cardboard or a wave appearance. And this really helps form the initial 3D structure of a protein. So there's strong hydrogen bonds again between adjacent chains. Now let's discuss the most important structure, which is the tertiary structure of proteins. The tertiary structure is the most important because it helps in determining the function of the protein. We have two pictures of different tertiary proteins on the slide. And as you can see by just the view of them, they are very complex. There's lots of interactions happening and they are folded. So when it comes down to proteins, folding gives them a function. And so this is the folding of the secondary structures into compact globular proteins. And the folding is really determined by interactions between side chains. I posted a link to a YouTube video that helps clarify this and makes it a little bit more clear of the transition between a DNA sequence, mRNA transcription, translation into a primary structure to a secondary structure, and now folding into a tertiary structure. So not all proteins get to this level. Some stay in a secondary structure, like your fingernails and your toenails. But many structures get to a tertiary because now we're starting to see proteins have a function within the body, specifically enzymes. So enzymes are usually found in at least a tertiary form. So, for example, we have two pictures here. We have superoxide dismutase, or SOD, and this is an important enzyme. It actually converts superoxide anion to a hydrogen peroxide, which is important in the body. We also on the right have catalase, which converts hydrogen peroxide to water, and this is also a very important enzyme to catalyze that reaction. So we see a lot of enzymes in the tertiary structure of proteins. There are four main forces that hold the tertiary structure together. These are the interactions. And so there's four main interactions or forces. The first one is disulfide bonds. And we can see this on the right-hand side. This is an interaction between two sulfur atoms on adjacent cysteine amino acids. So it only happens between sulfur and adjacent cysteine amino acids. The second one is hydrogen bonds, and these are between partial positive hydrogens and partial negative atoms of, for example, oxygen. So its polarity exists. So we create a polar relationship here, and that is of a hydrogen bond. We also have salt bridges, which are weak bonds between positive and negative side chains. So again, polars. And then the fourth one is hydrophobic interactions, which are interactions between two or more non-polar side chains. The key here is non-polar. So hydrogen bonds and salt bridges are interactions between poles, and they're polar, but hydrophobic interactions are interactions between two or more non-polar side chains. So the disulfide bonds are covalent. The hydrogen bond and the salt bridges, as well as the hydrophobic forces, are non-covalent. So a salt is formed by the neutralization reaction between acids and bases, and that's what we see there. So hair, as we mentioned before, is made up of protein, and the tertiary structure of hair is stabilized by the presence of disulfide bonds. The disulfide bonds will determine whether hair is straight, curly, or wavy. So the more disulfide bonds you have, the more curly your hair is. The less number of disulfide bonds you have, the more straight your hair is. So in chemical processing, these disulfide bonds are broken by something called reduction reactions. So when you add heat, when you're straightening your hair, that's chemical processing, you can actually break these disulfide bonds through the process of reduction. So then you lose that curliness and it becomes more straight. So you're actually disrupting the disulfide bonds. So when that happens, the hair is positioned in the new configuration and the disulfide bonds are reformed by oxidation. Hence the reason why when you straighten your hair, it will bounce back to its original state. And that's because it will stay in the new configuration, the straight way, for a little while, but the disulfide bonds are reformed by oxidation over time, so that straight hair becomes curly again. Also, another chemical processing is when we treat it with water, when we have a shower. That actually reforms through oxidation your disulfide bonds, hence the reason why your hair is usually more curly after a shower if you have that gene. Okay, so hydrogen bonds are also disrupted by heat and moisture, which then reform when the hair cools. So now let's talk about our hair a little bit more. So remember, hair is made up of protein. And by using a straightener, we actually disrupt the tertiary structure of hair protein. And genetically, we each are blessed with a certain amount of disulfide bonds. So if you have really curly hair, you have greater number of disulfide bonds than wavy hair, which is greater number than straight hair. And so through chemical processing, for example, through heat, when you go to straighten your hair, it actually is the process of reduction, where we're breaking the disulfide bonds. And then over time, whether you're using water or moisture, you actually cause an oxidation reaction to rebuild your disulfide bonds. So hydrogen bonds are also sensitive to heat and moisture, and the hydrogen bonds will reform when the hair actually cools. So if you have curly hair, you have more disulfide bonds, and if you have straight hair, you have less disulfide bonds. Now let's talk about our last and most complex protein structure, and this is our quaternary structure of proteins. And on the right is a nice figure outlining the primary, secondary, tertiary, and quaternary proteins, and it provides a really great summary that I highly recommend you look at because it will probably make a little bit more sense. So ultimately, we have our DNA. Our DNA codes for a specific mRNA sequence. That mRNA travels from the nucleus in the cell to the ribosome, which then codes for a specific polypeptide sequence. In the process of translation, it translates the RNA into an amino acid polypeptide sequence. That leads to our primary protein structure, which then, if it needs to, it will move into the secondary protein structure, the alpha helix or the beta pleated sheets. It then could potentially move into the tertiary structure where we see folding happening. And now the protein has a function, a biological function. Sometimes that protein, that tertiary structure, will move into a quaternary structure, but not all the time. Some just stay in the tertiary structure. Okay, so let's talk about these quaternary proteins. They're a combination of proteins in their tertiary structure. So if you look at the picture on the bottom right, we see subunits, and they're these round circles. Well, each of those round circles indicates a tertiary protein that are coming together and forming a quaternary protein. So each of those tertiary structures, proteins, are called subunits. So a quaternary protein is very complex because it's multiple tertiary proteins coming together to form a big molecule. So when the proteins do form a quaternary structure, the tertiary structure is called a subunit. And as such, it forms this multi-subunit complex, which is quite a large protein. It can be the same or different, these subunits. You can have multiple of the same subunits coming together or different ones. And it really allows for a huge possibility of different types of functional biological proteins within the body, because you can have any type of combination of tertiary proteins coming together. One caveat to all of this is that the subunits are held together by weak forces, specifically hydrophobic forces and H bonds, which are not that strong. And so they are easily broken and can go back to the tertiary form of these protein structures. Now let's look at one huge quaternary protein. So remember, the quaternary protein is made by multiple tertiary proteins, also known as subunits, coming together to form a very complex, large quaternary protein. So this picture on the left-hand side, we see multiple subunits that are dissociated. So this enzyme is actually inactive when all of the subunits are dissociated. But when these subunits come together and are associated, the enzyme becomes activated. And this enzyme is known as nicotinamide adenine dinucleotide phosphate oxidase. And it's a multi-subunit protein. It's the major producer of superoxide anion, which contributes to the major reactive oxygen species. And it must have all the subunits present for proper functioning of this enzyme. So this is a prime example of a quaternary protein with multiple subunits coming together to activate this enzyme. Now I wanted to discuss with you something a little bit more light in content. And this is the whole idea about drinking a raw egg. When I was a kid, I know my uncle was a bodybuilder and he would drink multiple raw eggs in the morning, thinking that it was complete protein, undenatured. It was the perfect source of protein, let alone the fact that you may have salmonella and some added bacteria and different things in it that could cause concern and havoc in your body. So what is the best way? Is it true that eating a raw egg is actually better for you than a cooked egg? Well, let's talk about this for a minute. So what happens with heating? Any chemical processing to a protein can denature certain bonds. So this egg, in its original form in the shell, may be in the amino acids, may be in a tertiary form. But with heating, and when you're frying the egg or cooking the egg, it actually breaks down the protein, essentially breaks down the bonds. So it could break down disulfide bonds, the hydrogen bonds, the hydrophobic bonds that form that tertiary protein. And so once that happens, it goes back to its secondary protein structure. With more heating, it will denature to its primary protein structure, which is that linear polypeptide amino acid sequence. And so even when you're cooking it, you're still getting a protein source and you're still getting those amino acids in the primary state. So it's not like the amino acids just disappear. No, the cooking process denatures the protein back to its primary form. So you're still getting those amino acids. So my uncle could have actually had the cooked egg and still be getting amino acids. It's just the protein structure that changes. The nutritional value is still there, and you're still getting the amino acids that your body needs to build, grow, and repair your own proteins. To end off this mini lecture today, we're going to do a learning check. So some questions that I want you to pause the recording right now and try to answer on your own. The first question is, what type of bond is responsible for the primary structure of proteins? What we went over today is called a peptide bond, which is covalent. So it's a covalent peptide bond that's responsible for the primary structure of proteins. Remember, the primary structure is a chain of repeated amino acids that are bound together by peptide bonds to form a large polypeptide. Let's move on to the second question. What type of bond is responsible for the secondary structure of proteins? We know this as hydrogen, non-covalent bonds. So these are hydrogen bonds that are between the amine and the carbonyl or carboxyl group of the repeated amino acids. What type of bond are responsible for the tertiary structure of proteins? There are four. The first one is a disulfide bond, which is a covalent bond. We have hydrogen bonds, which are non-covalent. We have salt bridges that are non-covalent. And we have hydrophobic interactions, which are also non-covalent. So there's four types of bonds responsible for the tertiary structure of proteins. The last question is, all proteins are characterized by what level of structure? Well, essentially, a protein has a function when it's in its tertiary form. So in this case, all proteins are characterized by the tertiary level of structure, because the tertiary level, the folding of an amino acid linear structure, gives it its function. So it's the tertiary level. This concludes this online lecture.